Assalamu alaikum warahmatullah. Welcome to the third lecture of carbohydrate metabolism. We will be discussing pentose phosphate pathway, metabolism of two other hexoses, fructose and galactose, in addition to uronic acid pathway. Our objectives today are to explain the two phases of pentose phosphate pathway and their significance, to identify the key enzymes of the pathway, and to explain the role of the pathway in production of NADPH and ribose 5-phosphate, and to discuss the role of NADPH in biosynthetic reactions and protection against oxidative stress and drug metabolism, and to discuss glucose 6-phosphate deficiency as it is related to the pathway, and to compare and contrast glycolysis and pentose phosphate pathway. We'll also discuss the key steps of fructose metabolism in the liver, and we are going to list the main examples of fructose metabolism to outline the process of galactose conversion to glucose 1-phosphate and UDP glucose, and to explain the importance of UDP galactose and UDP glucose and cellular function. We are going to list the main examples of galactose metabolism disorders, and discuss finally the role of uronic acid pathway in production of glucuronic acid, ascorbic acid, and pentoses. So let's go. Pentose phosphate pathway is also known as hexose monophosphate shunt and phosphogluconate pathway. Hexose monophosphate shunt because we are talking about hexose monophosphate phosphates, particularly we will be starting by glucose 6-phosphate as an initial substrate, and phosphogluconate is actually a metabolite also in the pathway, as we are going to see. In our body, oxidation of pathway proceeds through two major pathways that are glycolysis, we have already discussed it, and pentose phosphate pathway, we will discuss it today, in addition to uronic acid pathway. So when we define pentose phosphate pathway, we define it as an alternative route for oxidation of glucose. Alternative to what? Alternative to glycolysis, as a major pathway for glucose oxidation. However, pentose phosphate pathway is more complex than glycolysis, and it does not generate ATP like glycolysis does. The two main functions of pentose phosphate pathway are generation of NADPH, which is needed for reductive synthesis, meaning synthesis of steroids and fatty acids, in addition to prevention of oxidative damage. Pentose phosphate pathway also functions in production of ribose residues. These ribose sugars will be required for nucleotides and nucleic acid synthesis. The role of NADPH in detoxification, particularly in the red blood cell. Pentose phosphate pathway, as you can see here to your left, generates NADPH. NADPH is needed by glutathione reductase in addition to FAD to reduce glutathione. Reduced glutathione is needed by glutathione peroxidase in order to detoxify hydrogen peroxide into water. So if there is no function in pentose phosphate pathway, then will be no production of NADPH, and hydrogen peroxide will accumulate and lead to cellular dysfunction. Where does pentose phosphate pathway takes place? In many tissues, all of which are active in either reductive synthesis and or detoxification of drugs and metabolites. A major location is the liver, in addition to adipose tissue, adrenal cortex, thyroid hormones, erythrocytes, testes, and lactating mammary glands. In the skeletal muscles, the pentose pathway is present, however, it has low activity. We will discuss the peculiarities of pentose phosphate pathway in skeletal muscles shortly. The general layout of the pentose phosphate pathway is that it is not straightforward like glycolysis. It is not linear. However, it is multicyclic. We start with 3 molecules of glucose 6-phosphate. In glycolysis, our initial substrate is 1 molecule of glucose. But in the pentose phosphate pathway, for one cycle, we start by 3 molecules of glucose 6-phosphate, which will give rise to 2 molecules of carbon dioxide and 3 molecules of 5 carbon atom metabolites. The 5 carbon atom molecules will be rearranged to regenerate 2 molecules of glucose 6-phosphate and 1 molecule of glyceraldehyde 3-phosphate. Let us see a schematic representation here. We start with 3 molecules of glucose 6-phosphate. We generate 5 molecules of 5 carbon atom, 3 molecules of 5 carbon atom compounds, which will recombine to regenerate the 6 carbon atom compounds in addition to glyceraldehyde 3-phosphate. So we started with how many carbon atoms? 6 plus 6 plus 6, 18 carbon atoms. We are ending with 6 plus 6 plus 3, 15 carbon atoms. The 3 carbon atoms will be lost as carbon dioxide. Pentose phosphate pathway has 2 main phases: an oxidative non-reversible phase, in which the glucose 6-phosphate is dehydrogenated to ribulose 5-phosphate, followed by a non-oxidative reversible phase, in which ribulose 5-phosphate is converted back to glucose 6-phosphate and glyceraldehyde 3-phosphate by series of reactions. Let's see. The red line here demarcates the 2 phases. The first oxidative non-reversible phase, as you can see, the arrows are non-reversible, includes 2 reactions, and the rest are part of the second non-oxidative reversible phase of pentose phosphate pathway. Oxidative non-reversible phase of pentose phosphate pathway starts with oxidation. In fact, it is a dehydrogenation of glucose 6-phosphate to 6-phosphogluconate, followed by oxidative decarboxylation of 6-phosphogluconate to ribulose 5-phosphate. Glucose 6-phosphate is dehydrogenated to 6-phosphogluconate. NADP is reduced to NADPH. So NADP receives the hydrogen from glucose 6-phosphate, and NADP will be reduced while glucose 6-phosphate is oxidized to 6-phosphogluconate. A second reaction is oxidative decarboxylation with removal of carbon dioxide atom from each of the 3 glucose 6-phosphate and formation of ribulose 5-phosphate. Again, NADP is reduced to NADPH. This reaction is similar to our 3 initial substrates, which are 3 molecules of glucose 6-phosphate. The non-oxidative reversible phase, the second phase, ribulose 5-phosphate, is a substrate for 2 different enzymes, an epimerase and a ketoisomerase. The epimerase here, in the reaction to your right and the farmost and to your left also, so 2 molecules of ribulose 5-phosphate will be acted upon by an epimerase to generate xylulose 5-phosphate. The ribulose 5-phosphate in the middle will be acted upon by a ketoisomerase to form ribose 5-phosphate. This ribose 5-phosphate can directly join the synthetic pathway of nucleic acids and nucleotides. And then we have the action of 2 more enzymes, a transketolase and a transaldolase. A transketolase transfers 2 carbon units. A transaldolase transfers 3 carbon units. A transketolase here transfers 2 carbon units from this ribose 5-phosphate, adds it to xylulose 5-phosphate to form pseudoheptulose and a glyceraldehyde 3-phosphate. So a transketolase deduces 2 carbon atoms from a 5 carbon atom compound, adds it to the other 5 carbon atom substrate to form a 7 carbon atom substrate and a 3 carbon atom substrate. And then a transaldolase transfers 3 carbon atoms compounds and adds them to the glyceraldehyde from the pseudoheptulose to glyceraldehyde 3-phosphate to reform fructose 6-phosphate and erythrose 5-phosphate. When you deduce the 3 carbon atoms from pseudoheptulose, it will become a 4 carbon atom erythrose. The 3 carbon atoms are added to the glyceraldehyde 3-phosphate to form a 6 carbon atom fructose 6-phosphate. And then a transketolase again transfers 2 carbon atom in the form of glycoaldehyde to form xylulose 5 to form erythrose 5-phosphate and to reform a 6 carbon atom compound again. Here we have our fructose, we have our erythrose 4-phosphate in addition to the third xylulose 5-phosphate. A transketolase will transfer 2 carbon atoms from xylulose 5-phosphates, add them to erythrose 5-phosphate, form 6 carbon atom fructose 6-phosphate. The xylulose 5-phosphate at the reduction of the 2 carbon atoms becomes a 3 carbon atom glyceraldehyde 3-phosphate. The fructose 6-phosphate, of course, by the action of phosphohexose isomerase, you remember it from your previous reactions, interconverts 6-fructose 6-phosphate and glucose 6-phosphate. So we can isomerize fructose 6-phosphate to glucose 6-phosphate. So the end result so far from our initial 3 substrates, the 3 molecules of glucose 6-phosphate, we regenerated glucose 6-phosphate again, 2 molecules. One molecule of 3 carbon atom glyceraldehyde 3-phosphate, and the other 3 carbon atoms are lost as carbon dioxide in the 3 decarboxylation reactions in the first phase. So the overall reaction so far, if you want to summarize it, you start with 3 molecules of glucose 6-phosphate, you use 6 molecules of NADP, 3 carbon atoms are released as carbon dioxide in the carboxylation reaction. You regenerated 2 molecules of glucose 6-phosphate, 1 molecule of glyceraldehyde 3-phosphate, and you produced 6 molecules of NADPH. If you want to make sure if there is net gain or net loss of carbon atom, let us calculate them. 3 molecules of glucose 6-phosphate means 3 by 6, 18 carbon atoms. In the products, we have 3 carbon atoms as carbon dioxide. We have 2 by 6, 12 carbon atoms as 2 molecules of glucose 6-phosphate, and 3 carbon atoms as glyceraldehyde 3-phosphate. So 3 plus 12 plus 3 is 18. 18 in the substrate, 18 in the reduction and product side. There is no net gain or loss of carbon atoms. For complete oxidation of glucose to carbon dioxide via the pentose phosphate pathway, we need to further metabolize the glyceraldehyde 3-phosphate. And this requires the action of 3 glycolytic enzymes, the phosphotriose isomerase, the aldolase, and the phosphohexose isomerase, in addition to 1 enzyme from gluconeogenesis, and that is fructose 1,6-bisphosphatase. Let's see how does that take place. We need 2 more cycles of pentose phosphate pathway in order to convert the two molecules of glyceraldehyde 3-phosphate to one molecule of glucose 6-phosphate, and the overall reaction will be starting by 6. We said 2 cycles of pentose phosphate pathway. Each cycle has 3 molecules of glucose 6-phosphate. So we start by 6 by double the amount of glucose 6-phosphate. We need double the amount of NADPs, that is 12 NADPs, to release to completely oxidize one molecule of glucose 6-phosphate to 6 carbon dioxide molecules. We regenerate the other 5 carbon glucose 6-phosphate molecules with production of 12 NADPs. So these are the starting 6 molecules of glucose 6-phosphate. We regenerate 5 of them, and for half of glucose 6-phosphate will be combined with half of glucose 6-phosphate as a glyceraldehyde 3-phosphate to regenerate the 5th glucose 6-phosphate. The 6th glucose 6-phosphate, of course, is lost as carbon dioxide, as we said. Now we come to what is peculiar about pentose phosphate pathway in muscles. Ribose is not a significant constituent of systemic blood. So when you draw a blood sample, if you estimated the different sugars in that blood samples, ribose will not be that much. Therefore, tissues must satisfy their own need. Every cell must make its own ribose. It is not going to get it from the circulation, unlike glucose, for example, or fructose. Muscle tissue contain very small amounts of glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase. That is the 2 enzymes of the first phase of pentose phosphate pathway. Nevertheless, muscle can synthesize ribose 5-phosphate for nucleotide synthesis. How is that? How the muscles, while they do not contain the first 2 enzymes that are required for the pentose phosphate pathway, can still generate ribose phosphate for their nucleotide synthesis? And the answer is that muscles reverse the second phase, the non-oxidative phase of pentose phosphate pathway, and then it will not need to go through the first pathway for which it doesn't have the enzyme, which is brilliant. So in muscles, it starts from here, it goes back in the other direction. The reactions are reversible, ending up in the formation of xylulose phosphate pathway, a pentose that is needed for nucleotide synthesis. What is the role of pentose phosphate pathway in preventing oxidative damage? We have already discussed it briefly, but for red blood cells in particular, pentose phosphate pathway provides NADPH for reduction of oxidized glutathione, and as we said, we need it to reduce hydrogen peroxide to water. Accumulation of hydrogen peroxide decreases the lifespan of erythrocyte, leading to destruction of the RBCs, which will manifest as hemolysis or hemolytic anemia. Oxygen can generate several forms of free radicals, including superoxide radicals and hydrogen peroxide and hydroxyl free radical. If you want to break this cycle, we can do that through glutathione peroxidase, which converts hydrogen peroxide to water. However, glutathione peroxidase requires glutathione in the reduced form to use it as a source of reducing equivalence to reduce hydrogen peroxide to water. You need glutathione in the reduced form. Glutathione in the reduced form comes from reduction of oxidized glutathione. And the ultimate source of reducing glutathione is NADPH that is coming from the pentose phosphate pathway. Glucose 6-phosphate dehydrogenase deficiency. If the enzyme is deficient, this will lead to impairment of the production of NADPH with accumulation of reducing of hydrogen peroxide and free radicals, as we said, manifesting as hemolytic anemia. And this is particularly evident in susceptible individuals who have the deficiency and they have ingested or have been receiving oxidants such as primaquine, aspirin, sulfonamides, or parabens. So if a patient is diagnosed with this deficiency, you will counsel the patient as to avoid these medications and other things in his or her diet. Otherwise, he or she will have frequent episodes of hemolytic anemia that might necessitate hospitalization and blood transfusion. This is an exercise for you. I would like you to compare and contrast glycolysis regarding these items. Now we proceed to the metabolism of other hexoses, namely fructose and galactose. Quantitatively, the most important dietary hexoses are glucose, fructose, galactose. Regarding glucose, we have already discussed glycolysis, gluconeogenesis, and pentose phosphate pathway. So let us talk a little bit about fructose and galactose. Glucose is derived from dietary starch, of course, as you know, fructose from sucrose and galactose from lactose. In the body, we have specialized pathways, particularly in the liver, that are required for the conversion of fructose and galactose back to glucose. The significance of fructose metabolism stems from its role in energy production in glycolysis. Fructose is an intermediate in glycolysis, as you know, glycogen synthesis and fat synthesis, the formation of glucosamine, which are needed for formation of amino sugars. For the fetal circulation of seminal plasma. So fructose is actually the energy source for the fetus and for the sperm. Diets rich in sucrose or high fructose syrup in manufactured foods and beverages lead to large amounts of fructose and glucose entering the hepatic vein through the after absorption from the intestine. Fructose is more rapidly metabolized by the liver compared to glucose. Why? Because fructose bypasses the highly regulated glycolytic steps catalyzed by glucokinase and fructokinase. And this allows fructose to flood the hepatic pathway, leading to enhanced synthesis of fatty acids, esterification of fatty acids, production of very low density lipoproteins, and raised concentrations or production of triacylglycerols and LDL in serum. So fructose is potentially atherogenic by virtue of increasing fats in the circulation. Fructokinase transfers a phosphate from ATP to fructose to form fructose 1-phosphate. So fructose coming from the diet will be phosphorylated at position 1 by fructokinase. The phosphate group is coming from ATP. Alternatively, fructose can be acted upon by hexokinase. We said hexokinase is not specific for glucose, it acts on many hexoses. Hexokinase alternatively can phosphorylate also fructose, however, at position number 6. But which pathway will fructose take? Fructokinase, particularly the hepatic, renal, and intestinal fructokinase, hepatic fructokinase in particular, has a very low Km and a very high affinity of for for the substrate fructose. So fructose will be taken up or we will join the fate of phosphorylation by fructokinase at position 1. At the same time, fructokinase does not act on glucose. Its activity is not affected by fasting or insulin, and that is why fructose disappears from the blood of diabetic patients at a normal rate. So if you injected an individual with equal doses of fructose and glucose and this individual is diabetic, or for some reason there is impairment of insulin in his or her body, and you observe for how fast the cells take up glucose and fructose, which one will disappear first into circulation. In the absence of insulin, cells cannot take up glucose because glucose transporters are not functioning well, and phosphorylation for keeping or trapping glucose intracellularly also is not functioning well. So most of glucose will be left in the circulation, forming hyperglycemia, while fructose will be taken up directly by the high affinity enzyme fructokinase, which is not affected by the absence of insulin. The second step is splitting of fructose 1-phosphate by aldolase B, forming two trioses, glyceraldehyde, just glyceraldehyde, not phosphorylated, and dihydroxyacetone phosphate, similar to that of glycolysis and gluconeogenesis. And then a triokinase phosphorylate glyceraldehyde to glyceraldehyde 3-phosphate. The two trioses then can proceed back through glycolysis to be completely degraded into pyruvate or lactate, depending on whether glycolysis was aerobic or anaerobic, and finally, of course, the citric acid cycle. Or the two trioses can be combined back by an aldolase to form glucose, and this is the fate of the majority of fructose. Through this route, fructose can form glucose in our body. So, fructose 1-phosphate, which has been formed through fructokinase, can be reversibly split by aldolase into dihydroxyacetone phosphate and glyceraldehyde, which can be phosphorylated by a triokinase to form glyceraldehyde 3-phosphate. The two trioses can be interconverted, of course, by a triose isomerase and can be joined back by either aldolase A or B to form 3-form fructose 1,6-bisphosphate, which can goes back to form fructose 6-phosphate and fructose 6-phosphate and releasing glucose eventually. We have what we call the sorbitol or the polyol pathway. This pathway is not found in the liver. It forms fructose from glucose by the action of aldolase reductase, which reduces glucose to sorbitol and uses NADP as a source of as a coenzyme. A sorbitol dehydrogenase then oxidizes sorbitol to fructose. This reaction requires NADP. So the sorbitol pathway, by the action of sorbitol dehydrogenase and aldolase reductase can form glucose from fructose. This pathway, as we said, is not found in the liver. The biomedical relevance of fructose metabolism manifests in diabetic cataract, The activity of sorbitol pathway increases with increased glucose concentration, for example, in diabetes, and particularly in the lens, peripheral nerves, and renal glomeruli. And as you know, diabetic patients if their diabetes is not controlled, they are prone to develop complications as blindness or loss of vision, peripheral neuropathy, and renal impairment or renal failure. All of these due to toxicity by glucose. How is glucose toxic to these tissues? By formation of sorbitol. And what is wrong with sorbitol? Sorbitol does not diffuse easily through cell membrane. So once it is formed, it is going to accumulate, increasing the osmotic pressure and increasing the volume of fluids inside the cell, leading to swelling and cellular impairment of all cellular functions, and eventually will manifest grossly as tissue damage and organ damage. This is just to remind you the anatomical position of the cornea. It is a glass-like structure. It is avascular. It does not contain vessels. Normally, it is clear, and this is a cornea that is clouded by cataract. We have three more disorders that are related to fructose metabolism, which are hereditary fructose intolerance due to deficiency of aldolase B, essential fructoseuria, that is, we find fructose in urine due to deficiency of hepatic fructokinase. Both conditions are treated by restricting fructose in the liver. A fourth disorder is fructose 1,6-bisphosphatase deficiency. It can cause hypoglycemia as well as hyperuricemia. And I would like you to look why this deficiency can result in hypoglycemia despite the presence of high glycogen reserves and hyperuricemia as well. This is for you to find out. Regarding galactose metabolism, galactose is required for the formation of lactose, which is the sugar of milk, or glycolipids, which are needed for cerebrosides, proteoglycans, and glycoproteins. Remember the difference between proteoglycans and glycoprotein? I would like you to refresh your memory by going back to your basic chemistry courses in lipids, carbohydrates, and proteins. A galactokinase phosphorylates galactose at position 1, similar to fructokinase that phosphorylates fructose at position 1. And then a galactose 1-phosphate uridyl transferase. From the name, it is a transferase. So it transfers what? A uridyl moiety from uridine diphosphate glucose, UDP glucose, to galactose to form UDP galactose. UDP galactose is the active form of galactose. We said the active form of fructose is fructose 6-phosphate. Of glucose is glucose 6-phosphate. Of fructose is fructose 6-phosphate. The active form of acetyl is acetyl-CoA. The active form of galactose is UDP galactose. So galactose phosphorylated by galactokinase. The source of phosphate group is ATP at position 1, leading to the formation of galactose 1-phosphate acted upon by uridyl transferase, which transfers UDP moiety from UDP glucose to form UDP galactose and UDP 1-phosphate. They sort of exchange the UDP for 1,4-phosphate at position 1. We said galactose metabolism is relevant to synthesis of lactose, which is needed for the production of milk in active mammary glands in humans and other mammals. Lactase synthase catalyzes the condensation of UDP galactose with glucose to form lactose. The lactose itself can be synthesized in the body from glucose. The body doesn't have to take it in the diet. UDP galactose 4 epimerase interconverts UDP glucose and UDP galactose, the two substrates for the synthesis of lactose. We have two relevant disorders of galactose metabolism, which are galactosemia and cataract. Galactosemia is related to increased concentration of galactose in the blood due to inability to metabolize galactose and deficiency of galactokinase, iodidyl transferase, and 4-epimerase, the three enzymes we have just mentioned in relation to galactose metabolism. Cataract results similarly from what we call a galacticol pathway, which is similar to the sorbitol pathway, leading to osmotic damage into the cells of the eye, particularly of the cornea. And the last section of our discussion today is uronic acid pathway. Uronic acid pathway is an alternative hepatic pathway for oxidation of glucose, albeit amino 1, and like pentose phosphate pathway, uronic acid pathway does not generate ATP. Uronic acid pathway role is to convert glucose to glucuronic acid, which is needed for glucuronidation reactions, to ascorbic acid, vitamin C, and to pentosis, which will be joining pentose phosphate pathway of nucleic acid and nucleotides synthesis. Glucose 6-phosphate will be converted to glucose 1-phosphate by a phosphoglucomatase. This reaction, we will see it again when we discuss glycogenesis, glycogen synthesis. The second reaction is conversion of glucose 1-phosphate to uridine diphosphate glucose, which again, this reaction is repeated in glycogenesis. The third step is a two-step oxidation of uridine diphosphate by a dehydrogenase to form uridine diphosphate glucuronate with production of NADH. UDP glucuronate or UDP glucuronic acid is the active form of glucuronate. It is involved in glucuronation reactions for the formation of glycosaminoglycans and proteoglycans molecules in the body, and for good conjugation process before excretion of steroids, some drugs, and bilirubin. We will talk about glucuronidation reaction when we come to xenobiotics and its role in bilirubin synthesis when we talk about heme metabolism. Vitamin C synthesis needs L-gluconate as a precursor. But in primates, including humans and guinea pigs, gluconate is not available or cannot be formed. In the other animals that can synthesize vitamin C, they start by uridine diphosphate glucuronate, which will be converted to D-glucuronate with release of the uridine diphosphate. D-glucuronate and then will be reduced to L-gluconate, and the source of reducing equivalent of the hydrogen here is NADPH2. L-gluconate then will be dehydrated and then oxidized to form 2-keto-gluconatone, which will eventually lead to formation of ascorbic acid. This step here, which is catalyzed by L-gluconolactone oxidase, is blocked or absent or deficient in primates and guinea pig, and that is why we cannot synthesize vitamin C in our body. We have to take it in the diet. Generation of pentosis by uronic acid pathway is by the two steps we have already discussed of formation of UDP glucose and UDP glucuronic acid, and then glucuronic acid will be converted through several reaction to xylose 5-phosphate. Xylose 5-phosphate can either enter pentose phosphate pathway or it can be used in other cellular processes. This concludes our talk today. Thank you very much. See you next time.